And we can keep going with our preparation for exam number 2, which is coming up this Saturday. So at this point, when we were going through, we were kind of looking through our exam objectives for lymphatic. We've gotten into immune as well, so we were getting into talking about the different types of MHC molecules and kind of what cells we find them on, what they're used for in terms of what cells are actually reading them, if you want to consider it that way, right? Looking at those self-antigens and what they're used for. And then that's about where we left off. So at this point, we still have the same, our period right here to just prepare. So what I can do is we can continue with our exam objectives, kind of reviewing those bigger ones that might be possible short answer questions. But also I wanted to check in with you to make sure that you didn't have any other questions, kind of like driving questions like, oh, but I really need to know this, and so I want to make sure that we get those questions answered as well. So when you guys are here today, do you have something that we need to address and get clarified for you? Yeah?
There's one, this is in respiratory.
Okay.
Explain how CO2 is transported by the blood, and then explain the role of carbonic anhydrase and the other ones.
And the other stuff in terms of how. Okay. Yeah, so that'll be a good one. We know that that is, I think it's one of our highlighted ones as well. Okay. So for sure, if we continue on with this, which is my plan, we should be able to hit that one and get that one covered, because I do want to make sure for respiratory system that we hit on a lot of those key processes that we were talking about, those 4 key processes, so we understand those. So yes, we'll get that. Other things? Okay. Well, I mean, like usual, if you have something, a question or a clarification or whatever, at any time when we're going through things today, or if you think of something, just raise your hand and we'll address that too. Okay? Okay. So then what we'll do is we'll continue kind of marching through some of these exam objectives so that we can make sure we have solid answers laid out for each of these major topics. Okay, so where we were last time, we'd kind of gotten through here and MHC molecules, and then we're starting to look at some of these other ones. So we've gone through, again, first line of defense, second line of defense, third line of defense. We said how first line of defense was fairly simple. We listed some cells and molecules that could be used as examples of first line of defense or external, non-specific. We went through those details about, okay, what makes it specific, non-specific, what do those things mean, what does adaptive mean, etc. And then, of course, external and internal were fairly self-explanatory. Second line of defense, we went through those examples as well. Again, cells and molecules, or even processes. We did have a couple of processes with second line of defense where we talked about fever and we talked about inflammation. So we got that covered. And then third line of defense is where we really started getting into how the specific nature of third line of defense is looking in particular at these self-antigens and foreign antigens, but it's not just, is it part of us or is it a foreigner? It really does depend on what particular pathogen we're infected with at the time. So what virus it is, what particular strain of virus it is, or bacterium, or fungus, or parasite, or something like that, right? Because we know that we have, when we're making these different B and T cells, which are the primary cells of third line of defense, we know that we make millions and millions of slightly different variants of those T cells and B cells, so that individual T cells and B cells have different receptors from their neighbors. So they're able to match with a different type of pathogen than their neighbors. And so it's like having, again, kind of, if you will, a keychain of a million or more different keys. Each one is going to work on a different lock. So each specific B cell or T cell that has their unique receptor is only going to work on a particular pathogen that we get infected with. So part of that specific nature has to do with that. And then when we are infected with something, we have to find, just like looking for a particular key for a lock, we have to find which T cell or which B cell has the right receptor that matches up with that foreign antigen that then is going to be able to respond to it appropriately. Okay? Okay. So that's kind of where we were getting into. Some of those topics having to do with, again, foreign antigens, self-antigens, etc. So now what we can do is we can do a little bit of work with, again, our B cells, our T cells, and of course with the T cells, we found out that there are two main types of T cells that we were discussing, our helper T cells and our cytotoxic T cells. So for each one of those, we need to know, okay, how does it initially, once we have a particular T cell that potentially is going to match with this pathogen that we're infected with, how does it get activated? What does it do in response to that activation? And what weapons does it have to actually fight and kill off that thing that we're infected with, right? How does it get us well again? So we need to be able to explain that for helper T cells, for cytotoxic T cells, and for B cells, right? And of course, remember when I'm saying cells, those are all lymphocytes, right? It's T lymphocytes, B lymphocytes, etc. We just kind of get a little lax with our shorter names like that. Okay, so we can see that 14 has to do with our cell-mediated response. So that is going to be T cells. The 16 that we see there has to do with our humoral or antibody-mediated response. So that's going to have to do with B cells instead. Okay? So what I'm going to do is I'm going to jump back to our PowerPoint where we can have like a graphic in place to help us review how those things happen. So let's talk about, so start out with a cytotoxic T cell or a helper T cell. So in our PowerPoint, it's kind of like slide 46 or 48, something like that. So this one has to do with our helper T cells. So let me bump up on that. Okay, so what we have here is we see an example of a helper T cell, that round cell underneath, down here. We have our helper T cell that is being presented with a foreign antigen. Now, how is that being presented to it? So what we see is that a helper T cell, I'm going to circle that just to make sure, square that, just to make sure we're understanding that's our helper T cell. We're seeing a helper T cell that is being presented, a foreign antigen, by an antigen-presenting cell. So an antigen-presenting cell is another cell that is actually straddling the line between our second line of defense and our third line of defense. So an antigen-presenting cell, we said there are different examples of cells that can serve as an antigen-presenting cell. Do you guys remember any examples? So how is that antigen-presenting cell? We see that it is showing off with an MHC2 molecule. We see that it's showing off a foreign antigen that we see there in kind of purple in combination with its MHC2 molecule. Where did it get that foreign antigen that it's showing off? Yeah, it ate it, right? So an antigen-presenting cell is a phagocyte. Remember, phagocyte is one of our examples of cells from our second line of defense. So it's going along and eating things. So this could be a dendritic cell or a macrophage or even like a B cell. So there's a few different examples of cells that are phagocytes that can then, after they've destroyed that pathogen, they can display what they've eaten and show it off in order to help our third line of defense, to help our specific defenses instead. So our antigen-presenting cell is some sort of phagocyte there. It's chomped up a pathogen, and now it is showing part of that foreign antigen, and I'll write that on there, our foreign antigen, yeah, I'll just cover it up like that. So we can see foreign antigen is that purple one there. It's showing that on its surface. So the threat of that one individual pathogen has already been taken care of, right? We've eaten it up, we've digested it with phagosomes and lysosomes, enzymes and stuff like that, and we've eaten it up, we've digested it, but we've taken some of those foreign antigens that were on its cell surface and we're showing them off. Now, what it needs to do is it needs to show those off, but it also has to show off as self-antigen so that a T cell or some other defensive cell doesn't attack the antigen-presenting cell, thinking that it is a foreign cell, right? So what it does is we see that we have an MHC2 molecule. Now, MHC2 molecules, remember, are a self-antigen that's only found on antigen-presenting cells, okay? MHC1 molecules are found on almost all of our body cells. MHC2 molecules only found on antigen-presenting cells. And what we can see is that MHC2 molecules used by antigen-presenting cells are read by helper T cells. So a helper T cell is going to have a foreign antigen presented to it in an MHC2 molecule. So an MHC2 molecule and a helper T cell, there's always going to be a link there. That's the type of antigen that it is working to recognize through this presentation. Okay, so we see a helper T cell. It comes up to this antigen-presenting cell, and it uses this CD4 protein on the side to read, if you will, that MHC2 molecule, that self-antigen. So first it checks that, and it says, oh, okay, I see you're on my team. Next, it uses this T cell receptor down here that has a very unique shape to it. It uses this second receptor to say, hey, okay, I see that you're on my team, but are you sick? I'm going to check you out and make sure that you're okay. So I'm going to use this other receptor and see if there's any foreign antigen sticking or oozing out of you, right? So I'm going to use the second one to check you. So if there was no foreign antigen on there, if it was an antigen-presenting cell and hasn't eaten anything recently, then this helper T cell has gone up to it and checked in and say, hey, oh, I see you're on my team. I'm not going to attack you. I'm not going to generate an immune response, raise the alarm. Do you have anything to show me? No, you don't? Okay, I'm just going to go along, right? But if there is a foreign antigen from something that's eaten on there, now we're going to go through this process, which is called clonal selection. So remember, we have all these different types of helper T cells that have slightly different receptors on them. We have to go through, like keys in a lock, we have to see which helper T cell has the right shaped receptor that's going to fit that foreign antigen, okay? So we go through a period of time of trading them out, until we find the one that has the right shaped receptor that fits that foreign antigen. So once we find that, then, this is now the helper T cell that's going to get activated. Because it's like, whoa, 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 I got the right key. I got the right one. It fits this one. I can help you out with this. So instead of just having one activated helper T cell, we need a whole bunch of them, right, in order to generate an immune response. So then we go through this step of what's called proliferation, right? Proliferation is where we go through mitotic divisions. We make more clones of that one activated helper T cell, so that we have a lot of them that can help with this particular pathogen. So that's where we see on the left side here, we get this clone army being generated, right? But in addition, we also differentiate, right? We make a slightly different subset of helper T cells that are called memory helper T cells. So these are memory helper T cells. They're not going to go into the fight today. They have the right shaped receptor, right, because they're all clones, but we're going to keep those off. And those will be in our system long term, so that if we get infected with that specific pathogen again, those memory cells will recognize it very easily and generate a new army, a new response that's very rapid and very intense to get rid of it if we get infected with it again. Okay? So this kind of shows us our path of how a helper T cell, a specific helper T cell, gets activated by a particular foreign antigen shown by an antigen-presenting cell using the MHC2 molecule, and how it then generates this whole army of cells that are going to war right now and help with this infection, and cells that will be memory cells to protect us long-term instead. So then the final step of that story is, great, we have this whole army of specific helper T cells for that particular pathogen. What are they going to do? How are they going to be helping us get over it? Okay? Now for the helper T cells, their weapon, if you want, now it's a helper T cell, so they're only helping with the immune response. They're not actually attacking, right? They are helping, and what they release, their chemicals or their weapons that they release, are these things that are called cytokines. They're also termed interleukins. So you can see IL-2 is interleukin-2. and leukin is like leukocyte. So you can use the term interleukin or you can use the term cytokines, but these are stimulatory chemicals. And what those chemicals do is that helper T cell will release these stimulatory or kind of like cheerleading chemicals onto B cells that are trying to create an army, or cytotoxic T cells that are trying to create an army, and it gets that proliferation to occur much, much faster, right? So the helper T cells release cytokines or interleukins, you can use either term, to stimulate the proliferation of activated B cells or activated cytotoxic T cells. Now we know that the B cells and the cytotoxic T cells, which we're about to talk about, they do have weapons that are going to be able to kill off whatever we've been infected with. But if we only have a few of those, it's not going to be very effective. We want a whole clone army of those cytotoxic and B cells to fight off whatever we're infected with. And the stimulation from these cytokines, from the helper T cells, allows us to generate a really large army really fast. It stimulates that proliferation of cytotoxic T cells and B cells so we generate that army so that they can go into battle. Okay? So that's their story about how helper T cells will generate and help us get over a particular pathogen, right? So they are stimulating the proliferation of other types of cells like cytotoxic T cells and B cells, etc. And so if you don't have helper T cells doing their job, you don't get that army being created. Now you have only a few B cells or T cells, and how effective is that going to be, right? They're not going to be able to find all of the different pathogens and attack them, etc. We need a lot of them, okay? Okay, so that's helper T cells. Question on that? All right. So now, let's talk about our cytotoxic T cells instead. So cytotoxic T cells, when we look at this example picture over here on the right, it looks kind of like the same that we saw with helper T cells. So what's the differences then? So this is a cytotoxic T cell. This is a T lymphocyte that can actually kill, right? It produces toxins, poisons that can get rid of pathogens. But what we see in the upper right-hand corner there is we see now an inactive cytotoxic T cell, and it's going up and it's being presented with a foreign antigen from a different type of cell. So what we see here is this is an infected body cell. Okay, so we have a cytotoxic T cell that's not going up to an antigen-presenting cell. It is going along and inspecting cells of our own body, like our epithelial cells and mucous membranes, or even deeper, like different types of cells, like whether it's muscle cells or fibroblasts or something like that. These cytotoxic T cells are on patrol, going through our tissues and checking all of these different cells that make up our tissues to make sure that they are not cancerous or virus infected or something like that, right? So they're part of our security team that is going and looking at these cells of our body. Now, in most cases, when it goes up to inspect these cells, our cells are okay and they're healthy, right? So it comes up to it. And again, you can see now, here's a little receptor. Now, this is called the CD8 protein as opposed to CD4 because there's two parts, right? 4 plus 4 equals 8, right? So it has a CD8 protein on it that it goes up to check that MHC1 molecule. We said MHC1 molecules are basically, right, ID tags for all of our cells to say, hey, I belong here, I'm on your team, we're all good, okay? So it checks that first. It says, oh, okay, I see, you're belonging to us, you're part of us. But then it wants to use that other receptor to see if that cell is infected with something, okay? So cytotoxic T cells are going to be most effective looking at our own body cells to see if they have some sort of some sort of within cell pathogen, right? So it's one of our own cells, but it's now become infected with like a virus or something like that. So it is going to be checking for that. So if it comes up to a healthy body cell, it uses the CD8 protein and goes, oh, I see you're on my team. And then it uses that other receptor to see if it's sick, right? If it's not sick, great. We're just going to pass on by, you're good, right? Just like a security guard might do. You're okay, we're going to pass by. But if that cell is infected with something, if it has these diseased agents inside of it, then part of those foreign antigens are going to show up on its cell surface as part of that MHC1 molecule. And it's going to be presented or displayed to the cytotoxic T cell. It says, oh, look at, I have all this inside of me. Please kill me off. Okay? All right, so just like usual, that cytotoxic T cell has to have a receptor that has a complementary shape to that specific antigen, right? And we go through that whole clonal selection again to find out which of our cytotoxic T cells has the right shaped receptor. Once we find the one that has the complementary shape, now we need a whole army of that particular cytotoxic T cell. So we see that same step, proliferation. We need tons of that particular one that's going to help us with that pathogen that we're infected with. So we see proliferation, lots of them made, and differentiation, where we have memory cytotoxic T cells being made and a whole clone army of cytotoxic T cells that are going to go into battle right then. Okay? So that again shows us our activation, how our cytotoxic T cells find their specific pathogen that's their whole life has been waiting for this, right? I've been waiting for this particular pathogen. I know how to deal with this one. My receptor fits this one. Now the question is, okay, memory cells, we'll keep those around in case we get infected with it again. How are those clone army cells of cytotoxic type, how are those going to defend us? What are their weapons? How are they going to poison these cells that have become infected, right? So that part of the story then, and this just goes through again, the types of targets that our cytotoxic T cells are commonly attacking. So that goes into then, what are these cytotoxic T cells? What are their weapons? So these are basically their weapons. So we have things like granzymes. These are chemicals that they release that trigger apoptosis. Basically like a bomb, a time bomb, throw it into that infected cell, and it tells that cell, you need to commit suicide. You need to kill yourself off because you're a goner, you're infected with this virus, and we don't want the virus to spread into other cells, okay? So we have granzymes, we have perforins as well. So perforins are going to create little perforations, and that's what we see here, right? So we can see here's a cytotoxic T cell that has bound with an infected body cell, and we are releasing from these granules these little granzymes and perforins. And we can see how the perforins form these little perforations, these little holes in the plasma membrane of our target cell. And it allows some of these other chemicals, like the granzymes, to get inside and start degrading the cell, eating it up, or actually causing it to go into apoptosis, causing it to die off. Okay? So those are the weapons that a cytotoxic T cell has once it's become activated, and once it identifies cells that are infected, those are the weapons that it has in order to kill it off. That's how a cytotoxic T cell is going to get you better from whatever you're infected with. Okay? All right, so we've had helper T cells, we have had cytotoxic T cells. We've been able to explain, again, how they identify some sort of pathogen originally, how they get activated, they make this clone army, as well as memory cells. That's a key aspect of third line of defense, is that we have this long-term protection from it, right, with it, and then the specific way that they protect the body. Okay, so now let's take a look at our B cells, our B lymphocytes instead. And so for that one, I think this figure, it's a little bit different than the ones that we were seeing before, but it works the same. What we see in this one is up at the top, we see, in this case, a few different examples. They're showing us a few different types of B cells, and they have slightly different receptors. We keep saying that we have all these different variants of B cells and T cells that have slightly different receptors. So what we can see is that this is a B cell with receptors on it. It's sitting in like lymphatic tissue. So it's sitting in a Peyer's patch. It's sitting in a tonsil. It's sitting in a lymph node. And it's getting all this material brought to it in general. So a lot of times, B cells are getting things brought to it as opposed to going out and inspecting the body like we see some of our T cells doing. So what we have is, here's a B cell, and what has been brought to it, a bunch of B cells in it, what has been brought to it is this specific antigen. And this antigen, this round antigen, happens to fit well with the kind of roundish complementary shaped receptors of this middle B cell here. So this one has more of a V-shaped receptor. This receptor is like an inverted V. Doesn't fit with it. It definitely does fit with this one instead. So that's the particular B cell that is going to be effective against this pathogen. So what do we need to do? Like usual, we're going to create a clone army that is going to go into battle. And then in addition, we're going to create some memory cells, some memory B cells. Now the clone army that we create, once this B cell has become activated, they actually have a specific name this time. They are called plasma cells. So plasma cells are activated B cells that have swollen up, gotten really big, and they're cranking out antibodies. Because antibodies are the chemical weapon, if we want to consider that, of the B cells. That's what they're using to help us get better, help us get over a particular infection with a pathogen. And we can see that those plasma cells are releasing those antibodies. So these happen to be little Y-shaped antibodies like that. We can see that they get much bigger because they have more rough endoplasmic reticulum, which is the machinery that creates these proteins to be released. And these antibodies then just get released into the fluids, like into the lymph. And we know the lymph goes back into the cardiovascular system, goes back into the blood. So now antibodies are in the blood. These antibodies sometimes can get across some of our different membranes as well, some of the walls of vessels and things like that. So they can also get into some of our other bodily fluids. That's also how we talked about a long time ago, how sometimes antibodies can be passed on to a baby, right? So those antibodies can cross across the placenta. They can get into breast milk and then be transferred to the baby that way. So these antibodies are pretty small. They're molecules, and they just flood into the fluids of the body. That's why it's called humoral response or antibody-mediated response. A humor is a fluid, okay? All right. So now what we see is, all right, so we saw a B cell getting activated, usually in lymphatic tissue, waiting for something to be brought to it. We created these plasma cells as well as our memory B cells, and these plasma cells start cranking out antibodies. So great, now we have these antibodies. What are these antibodies going to do? So now that's what we need to discuss. What are these antibodies? How are they used as weapons? The only bottom thing that we see, thing that we see down towards the bottom part of the slide is we see what's called a secondary response as well. So a primary response is when we first encounter a particular pathogen. A secondary response is always going to come from our memory cells. So our memory cells are what are going to actually be able to generate this secondary response because they have seen this pathogen before. We have a number of these circulating around, these memory B cells, and if they encounter that same pathogen again, they very quickly can produce a new clone army that then start releasing all of those antibodies again into the fluids. So it's a much faster response and a much more intense response on that second or third or fourth response when you get infected with that particular pathogen again. Okay? So all the time, that memory we get, that long-term protection, when we get exposed the second time, the third time, or whatever, it comes from our memory cells. And that's why through time, this third line of defense becomes more and more powerful, becomes better and better because it remembers every single thing that you've been infected with previously so that it will protect you again from that in the future. Okay? It's kind of like you guys, right? As you go through and you learn classes, as you learn content in classes, your brains get bigger and bigger as well. You're learning more material, so you become more effective in different ways as well, right? So you're changing. What you know now is a whole lot more than what you knew when you were 7 years old, right? So you are also changing. Our adaptive immune system changes because of its memory as well. Okay, so let's then talk about what happens in terms of how those antibodies protect us. That's this slide. So this shows us different specific ways that antibodies work to help get rid of a particular pathogen. So we see one here, neutralization. So neutralization is where these antibodies coat toxins or even free viruses that are floating around on their own. They coat them so that they can not affect our cells, right? So they're basically kind of wrapping them in antibodies so that they can't sink into and get inside of our cells and infect new cells or affect cells if we're talking about a toxin. So we're neutralizing the threat, okay? So that's neutralization. Agglutination is another way that antibodies can help us, in that this is what I talked about as like molecular handcuffs, where they will bind to, they have multiple binding pockets, these antibodies, they will bind to multiple cells or multiple antigens and bring them all together and hold them in one place, so that if we have a big macrophage coming along, they're going to be able to ingest and get rid of those all at once, rather than having to track down individual ones, right? So agglutination or clumping, like we saw even during our blood typing, is an important benefit of antibodies as well. Now, we've been showing it at the top there. There are different types of, different classes of antibodies, IgA and IgG and all this type of thing. Some of them only have a couple of binding pockets like this, 2 binding pockets. But you can see in this case, this type of antibody has actually 10 different binding pockets, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it can bind 10 different things in those pockets and hold them all in one place. So that's pretty effective as well. Third thing is precipitation. So precipitation is like rain, right? So where we have soluble antigens that are in a fluid and they're kind of hard to locate again, they're kind of floating around in the fluid. When we stick antibodies to them, they drop out of solution. So they settle out, right? Like a chemistry experiment, you get a precipitant that settles out of solution. And now that it's kind of bumbling around on the bottom, it's much easier again for different phagocytes to come along and eat up and get rid of, rather than, again, having it all floating around. So they can precipitate or cause these soluble antigens to come out of solution as one big clump. They also can help to activate the complement proteins. So complement proteins assemble a membrane attack complex that create these big holes in the plasma membrane of cells. And if there are antibodies sticking to a cell, that actually can initiate the complement proteins creating their membrane attack complex. So they work to help initiate or work with the complement proteins to poke holes in that cell as well, okay? So they work with the complement proteins. So that's a fourth way that they kind of enhance it. A fifth way is they do opsonization. So opsonization is when they're sticking, like you see kind of maybe over here, when they're sticking off of a particular pathogen, they act like little flags, right? They flag that pathogen, and now our phagocyte, as it's cruising around trying to find things to eat, it sees all these flags sticking off, making it much more obvious, and those antibodies work as little grips or handholds that it can reach out with its pseudopods, grab onto, and it's more easily able to ingest it and phagocytize it. So opsonization is where our pathogens are flagged with antibodies, and they act as opsonins, making it easier for the macrophage or the phagocyte to ingest it. So they kind of give it handholds, if you will, or the ability to identify and hold on to that antigen a little bit easier. It makes it easier to eat. So these are the different ways. These are different examples of how antibodies produced by B cells will get us well. Okay? So again, like when we talk about these, you don't have to know every single way, but you need to have examples, right? So if we're explaining how B cells work and how B cells get activated and what they do to protect us, you've got to be able to describe that in detail if you get it like as a short answer question. Okay? Okay. And then, of course, you can kind of work backwards with this information, too. If you're thinking about a multiple choice question, you could have one of these terms in terms of agglutination or neutralization or something like that, have a description of it, and then be able to pick it out. So if we say, clumping of antigens by antibodies is called neutralization, precipitation, agglutination, opsonization, you'd be able to kind of pick it out that way as well. Okay. So For third line of defense then, what we've gone through in detail is we've talked about our three major types of cells, our cytotoxic T cells, our helper T cells, and our B cells. How they get activated, what they do in response, and how their secretions, how their chemicals that they release will help to get us better. Okay? And I think, now if we go back to this and we look at it, we've been able to explain a lot of what we see here, right? So we talked about helper T cells and their role in terms of the B cell response, as well as their role in terms of the T cell response, releasing their interleukins or their cytokines onto them so that they can produce more intense proliferation. We talked about cytotoxic T cells and what they do. There's some examples of some of their weapons, like we talked about, like perforins, etc. And then we talked about memory T cells and memory B cells as well. And then we talked about the antibodies being produced by plasma cells and what those antibodies do. And that kind of comes into this question, right? So what antibodies are and how they help to fight infections. Okay, so the only other thing that we have in there that maybe we didn't mention yet is immune surveillance. And immune surveillance is where our cytotoxic T cells, we said that they are going around the body and inspecting our body cells to see if they're healthy or not. Number one, do they belong with us or not? And number two, once they've identified them as being part of us, are they healthy or are they sick? And do we need to attack them and get rid of them, right? So that is part of immunosurveillance. Now, cytotoxic T cells would do that, but natural killer cells, as part of our second line of defense, they're a cousin to our cytotoxic T cells. Those also do that type of patrol where they're looking at all of our individual cells, making up our tissues, and seeing if they're healthy or not. Now, with cytotoxic T cells, it's going to take a very specific one that's going to have the right shaped receptor to generate that immune response. But for natural killer cell, it's more like, okay, are you on our team or are you not? If you're not, okay, I'm just going to kill you. And they have some of the same weapons. Natural killer cells have some of the same weapons as our cytotoxic, perforins and stuff like that. Okay? Okay, so both of those, the natural killer cells and the cytotoxic T cells, are doing that type of monitoring or surveillance of our body tissues to make sure that we're okay. So we call that immune surveillance. So that's what that is. Okay, so we already explained about the secondary response. That's coming from our memory cells and why it's very vigorous like that. And that takes us into respiratory system. People okay moving into respiratory system? Okay. So let's take a look at that. So if we look at the ones that are in bold of the respiratory system, again, I kind of separated this one. I'm not exactly sure why, just because sometimes it can be easier to look at the anatomy separate from the physiology when we look at just kind of, hey, what's this part? What's that part? What's that part? But of course, there's going to be overlap between the two. We always say that the anatomy goes hand in hand with the physiology, allows us to do a particular function because of the shape or the structure of that particular tissue or organ or whatever. So when we look through these bolded terms here, many of those bolded terms or bolded exam objectives have to do with our 4 different processes. Okay? So if you remember, our 4 different processes were, what's first step for respiratory system? Yeah, so the first step is ventilation. Ventilation or breathing. So that's our first step. We have to be able to explain how ventilation works. Okay, second step is after we take that air in, we've got to be able to exchange the oxygen and carbon dioxide. We want to get more oxygen into the blood. We want to get carbon dioxide out of the blood. So that is called external respiration. So we've got to be able to explain that, how that works. Third step, we now have oxygen in the blood, and we need to move it around to get it to the tissues that need it. So third step is gas transport. And then once we get it down to the tissues, I always think of a distant tissue, right? Like the bottom, a muscle in the bottom of your foot or something like that. Once we get it there, it's still in the bloodstream. We've got to be able to get that oxygen out of the bloodstream to the muscle cells or something like that. As well, we want to pick up some of their garbage. We want to pick up some of their carbon dioxide, transport that away. So in that case, our fourth step is going to be internal respiration, internal gas exchange. And as we were mentioning, when we say respiration, we're talking about that gas exchange. It's just whether it's with the external environment or whether it's in the internal environment. So in this case, this is going to be between, we'll write it on there, the alveoli. I'm going to separate that alveoli, don't forget that L. Alveoli and the pulmonary capillaries. So the capillaries surrounding the alveoli in the lung. I should probably put that as an IES. Okay? That's where that's occurring. Whereas internal respiration is occurring between the systemic capillaries and the tissues. Or just simply say the tissues. Okay? So that's a key aspect of when we're explaining external and internal respiration, is we do need to say where this is occurring, right? Because that's a different step, external versus internal respiration. Okay, so let's take a look at maybe some graphics from our PowerPoint handout that may help to explain each of those. So the first one that we were talking about is ventilation. So ventilation is going to be explained most readily by talking about the muscles that drive it and pressures. In fact, there are 3 different pressures that we have to be able to describe when we're talking about ventilation. So those 3 pressures are the ones that you see circled on the screen right there. Atmospheric pressure, intrapleural pressure, and intrapulmonary pressure. Sometimes intrapulmonary pressure is also called intraalveolar pressure because it's this pressure within the lungs itself. Okay, so we would have to list, if we're explaining ventilation, if you have a short answer question that says, talk to me about ventilation, tell me how it works, you've got to, number 1, you've got to list the muscles involved and what they're doing, and you have to talk about those 3 pressures. You have to list them and you have to say, why are those important? Okay? So first step, if you can list them, that's going to be good. You're going to get your partial credit, but you've got to explain them. Why are they important? So we said that intrapleural pressure, which is occurring in the pleural cavity, right, between the wall of the lung and the wall of the cavity, is creating that suction, sticking the wall of the lung to the wall of the cavity, so that when the muscles move the wall of the cavity, the lungs are going to follow suit. They're going to be stuck to it and they're going to follow suit. So we can see those numbers, right? These are pressure numbers. It says 756 millimeters of mercury. So remember that those are your units for pressure, millimeters of mercury. And we can see that intrapleural pressure is below intrapulmonary pressure, technically below atmospheric pressure as well. So that negative pressure, if you will, creates that suction so that it stays against the wall. That's your explanation for why intrapleural pressure is important. It keeps the wall of the lung stuck to the wall of the cavity. Okay. Next, we have to explain atmospheric pressure and intrapulmonary pressure. Now, in this case, we're just talking about pressure. Remember that pressure refers to all of these different molecules that make up air in a given volume. Okay? Now, there's lots of different molecules that make up air. Nitrogen, oxygen, carbon dioxide, carbon monoxide, water vapor, etc. So all those things packed into a particular volume give us a pressure. Okay? And so what we're looking to do is we're looking to move air. If we're doing inhalation or inspiration, we're looking at moving air from here into here, right? So we're looking at moving air from where atmospheric pressure is outside the body to inside of the lung. And the way we do that is that we are going to use this relationship between volume and pressure. And so we said that there's this inverse relationship between volume and pressure. The muscles that we have that are respiratory muscles are going to change the volume of that cavity. And so the muscles that are primarily involved there are going to be what? Do you remember? The main one starts with a D. diaphragm, right? So diaphragm, we're going to R-A-G-M like that. The diaphragm is the primary muscle for ventilation of the lungs. And then there's also the external intercostals, at least for resting breathing. So those are the muscles between the ribs, right? So you at least have to list those two muscles as well for causing us to breathe in and out. So when the diaphragm and the intercostal muscles contract, what's going to happen is we're going to make that volume bigger. So the walls of the cavity are going to come out here, and the diaphragm is going to flatten, and it's going to drop the floor, and it's going to make the cavity bigger this way as well. So we've just increased it both directions. So when those muscles contract, the volume gets bigger, and the pressure, the intrapulmonary pressure, goes down, right? Because they're inversely related. Once that intrapulmonary pressure is lower than atmospheric pressure, then the air will move from where it's high pressure to where it's low pressure, and it'll go into the lungs. Okay? So for instance, right now, we're saying this is like 760 millimeters of mercury, or 1 atmosphere. It's basically the same as atmospheric, so we wouldn't see any movement going on. But once we open up that volume a little bit more, we're going to see a drop in that pressure. So even if it goes down to 759 or 758, that's enough. Now we have low pressure inside of the lungs compared to out here, so the air is going to move in. So that's going to be our inhalation, where we have dropped the pressure inside of the lungs, in that intrapulmonary pressure. And then exhalation, where we're getting it to move the other way, we contracted these muscles and it made the volume bigger like this. Now we relax those muscles and everything comes in again, right? The side walls come in, the floor comes up again, and so that volume gets smaller, which drives the pressure up. Once the pressure in the lungs is higher than out here, now we have a gradient going the other way, the air will leave. It will go from high in the lungs here to where it's relatively low outside the body, and we will exhale instead. Okay? So with those, you have to be able to, again, talk about the diaphragm and the external intercostals, be able to explain when they contract kind of what they're doing. Again, the diaphragm flattens, and so that's going to create more space. And the walls of, when the external intercostals contract, the walls of the pulmonary cavity are going to expand laterally, is what we'd say, but to the side, right? So we're going to get expansion that way. When they relax, then everything's going to come back in. The diaphragm's going to raise. It's going to get more of that curved shape as opposed to flattened. And the walls of that thoracic cage are going to come down again. Okay? So what we're seeing is that those muscles change the volume, which changes the pressure inside the intrapulmonary pressure, right? And whether it's higher than or whether it's lower than atmospheric pressure drives the movement either into or out of the lungs. So atmospheric pressure doesn't really change, but we have to change the pressure inside the lungs relative to it to get the air to move one way or the other. Okay? Questions on that? Yeah?
The two pressures, intrapleural pressure and intrapulmonary.
Pulmonary, yeah?
What are the functions again?
So intrapleural pressure is sticking the wall of the lung to the wall of the cavity. Intrapulmonary pressure, that's the pressure that we have to change to make it lower than or higher than atmospheric to get the air to move into and out of the lung.
Okay. So is the wall of the lung sticking to what exactly?
To the wall of the cavity,
The wall of the cavity.
of the pleural cavity. That's for intrapleural pressure, because it's called the pleural cavity. So that's where you have your hook there, right? Pleural, it says intrapleural pressure, and pleural cavity.
Okay, thank you.
Mm-hmm. Okay, so that's ventilation. We're kind of using that diagram to explain ventilation. And then we need to talk a little bit about exchange, right? External and internal respiration. So this figure is showing us kind of that circulation, right? So it's showing us up here. It's showing us basically external respiration up here. And down here, it's showing us internal respiration. because you can see what are involved in these two spaces here. In these two spaces, we see gas exchange between the alveoli and the systemic capillaries. They call them alveolar capillaries, but pulmonary capillaries. And then down here, we see exchange between the systemic capillaries and the tissues, right? Okay. Now here, when we're talking about gas exchange, we're not talking about pressure overall. That was ventilation. Pressure overall is just like getting this big mass of air to move into the lungs or getting to move out of the lungs. Now, when we're talking about gas exchange, it's coming down to what are called partial pressures. We want to know what's the concentration or partial pressure of oxygen and CO2. We're interested in those two gases. We want to move those two gases. How do we get those two gases to go across the capillary wall to either go into or out of the alveolus or into or out of the tissue? How do we get them to move across that respiratory membrane or move across down at the tissues? So that has to do with partial pressures. So when we look at this graph, you see PO2 like this and PCO2 like that. Those are referring to partial pressures. It's saying, what's the concentration? That's what it translates to. What's the concentration or partial pressure of that molecule in this area versus this area? So we're still seeing gradients here, but we're talking about a concentration gradient or a partial pressure gradient of oxygen and carbon dioxide by themselves, okay? So in this area, when we're talking about external respiration, we see it written out much better than how I wrote it out right there. So in external respiration, what we're doing is we want to see where is oxygen high concentration, where is it low? Where is carbon dioxide high, where is it low? So for instance, we're considering what's the concentration of carbon dioxide, for instance, in this pulmonary capillary here. So in the pulmonary capillary, we have a relatively high partial pressure of carbon dioxide. It's about 45. In the alveolus, in this freshly inhaled air, the partial pressure or concentration of carbon dioxide is 40 instead. So we have a relatively, again, high concentration of carbon dioxide here and a relatively low concentration of carbon dioxide here. So carbon dioxide diffuses out of the capillaries from high to where it's low. Okay? Oxygen is going to do the same thing. And I'm realizing that I reversed my color scheme from the last time we went through this. But oxygen, let's look at its concentration gradient. So when we inhale air, we have a relatively high concentration of oxygen there, 104 millimeters of mercury. If we look at it in those alveolar or pulmonary capillaries, we see the concentration of oxygen is fairly low in this blue blood, right? So it's high concentration of oxygen here, it's low concentration of oxygen there, so oxygen diffuses into those pulmonary capillaries, okay? And then the same thing happens when we get to the tissues, but it's just in reverse. Here, oxygen levels are high in that arterial blood, relatively low in our active tissues, so oxygen is going to diffuse out, whereas carbon dioxide is going to be the opposite. In our active tissues, they are producing lots of carbon dioxide, so it's relatively high. It's relatively low here in this freshly oxygenated blood, and so carbon dioxide diffuses into the capillaries there for transport back to the heart and then back to the lungs, okay? So with this, what we're really focusing on in this diagram are those two interaction places between these two sets of capillaries, the pulmonary capillaries in the alveoli and the systemic capillaries in those tissues. So that's where we're looking at the exchange, and the exchange is due to the partial pressure or the concentration of each of those gases. Where is it high and where is it low? Now you don't have to worry about numbers. I'm not going to ask you where's it 140, what is the specific partial pressure of it here or there. You just need to be able to describe where it's relatively high and where is it low, okay? So like we saw, for oxygen in freshly inhaled air, oxygen partial pressure is going to be higher in the alveolus compared to those pulmonary capillaries. So then it's going to go from high to low, it's going to go into those pulmonary capillaries, right? So in each case, we just need you to say, where is it high and where is it low? And then it's going to diffuse from high to low. It's going to move that direction. Okay? So this is describing both external and internal respiration. So for external and internal respiration, you have to say where is it occurring, between what capillaries and then what tissues. And then you have to explain, okay, we're talking about partial pressure here, not overall pressure, just partial pressure, individual concentrations of oxygen and carbon dioxide, which is going to drive the movement of those two gases, in fact, opposite directions. So you're going to be able to explain that. Okay? Questions on that? Yeah. So in the tissues, what we're looking at down here is if we look at a tissue, if we expect a tissue that's actively using ATP, making and using ATP, right? We said that the mitochondria use aerobic respiration to make ATP. They utilize oxygen in that process, and at the end of it, they give away carbon dioxide. So in an active tissue, we're going to find that there's going to be relatively low partial pressure of oxygen because they're using it up, and it's going to have a relatively high partial pressure of carbon dioxide because it's actually producing that as a byproduct, right? So in the tissues, they have a relatively low concentration or partial pressure of oxygen and a relatively high partial pressure of carbon dioxide because they're working. So we're comparing that to that fresh arterial blood that's coming in, which has a relatively high partial pressure of oxygen, a nice high load of oxygen, and a relatively low amount of carbon dioxide because we got rid of it up in the lungs. Okay. So that is explaining our respiration that we're talking about there. Now, there was one question. It's coming up in terms of gas transport. Okay, so gas transport is our last topic that we see there. And gas transport, we have to, again, specifically discuss carbon dioxide and oxygen. How is it transported in the blood? So for oxygen is mainly transported bound to hemoglobin, right? So we said about 98% of it is bound to hemoglobin, about 2% of it just floats along in the plasma itself. And with the hemoglobin molecule in the red blood cells, we have 4 different binding pockets, 4 different heme groups with iron associated with them, where the oxygen is going to bind. And we said that based on the environment where that hemoglobin is traveling through at the time, it will either grab onto and load up with oxygen into those binding pockets, or it will be stimulated to release that oxygen instead, okay? So it reversibly binds with oxygen. It just has to know when's a good time to load up and when's a good time to release. This hemoglobin saturation curve that we see right here, you're not going to be asked to reproduce it, but you do need to know kind of what it means overall. This is actually showing that for saturation of hemoglobin, right? So we see 0% saturated, it's empty. 100% saturated, all 4 binding pockets are full with oxygen, right? So we're looking at the response in terms of the saturation of hemoglobin, in this case based on the concentration or partial pressure of oxygen in the environment that it's passing through. So is it passing through the lungs where there's a lot of oxygen? Is it passing through those active tissues where there's not a lot of oxygen, where there's a low concentration of oxygen, etc.? And what does it do? How does it respond? So what we see is that when we get to tissues like the lungs where there's a lot of oxygen around, that's like going to Costco for hemoglobin. It says, oh, okay, I got some empty binding pockets. I'm going to stuff my pockets with oxygen. And we see the saturation go up and up. So when it's passing through an area like the lungs where we have a high partial pressure of oxygen, it loads up on oxygen. But when it passes into a neighborhood where it's poor oxygen, lower concentrations of oxygen, then it adds the stimulation to it. It says, oh, okay, you're really poor here. Okay, I have lots. Here, have some of mine. Here you go, here you go. And it starts unloading. It starts becoming less and less saturated, right? More and more open pockets because it's giving it to that poor neighborhood, okay? So that's what that saturation curve shows, is that in essence, for partial pressure of oxygen, a high partial pressure of oxygen will cause it to become more saturated and it picks up oxygen. A low partial pressure or neighborhood that has a low concentration of oxygen, it'll be stimulatory, so it'll release oxygen instead. That's its response, okay? Now, it'll mainly respond to the concentration or partial pressure of oxygen in its neighborhoods going through, but it'll also respond to other factors like changes in temperature, changes in partial pressure of carbon dioxide as well. Now, again, you don't have to reproduce these curves or anything like that, but think about for each of these factors, if that factor goes up or goes down, what's the response for hemoglobin? Is it going to load up with oxygen, hold on to it, or is it going to release it? And as I was talking about, I like to think about an active tissue. If it's really active, like a muscle tissue, what's its environment going to be? Its environment is going to be low concentration of oxygen because it's using it to make that ATP. It's going to have a high partial pressure or concentration of carbon dioxide because those cells are working and cranking out carbon dioxide. It's going to be warm, right? Warm muscles, so it's going to be warmer. And it tends to be a little bit more acidic in that environment because, number one, it's making lactic acid. But number two, when it puts out carbon dioxide, we know, we're just about to review it, that carbon dioxide, the main way carbon dioxide gets transported is as bicarbonate ion. And when we convert it to bicarbonate ion, that releases a hydrogen ion. So it becomes more acidic. Every time carbon dioxide is released, the environment becomes more acidic. So what we're showing with that is that, for instance, when temperature goes up, our saturation, our hemoglobin saturation goes down because it's releasing that oxygen to that warm tissue that's working hard. So that's the relationship there. As carbon dioxide levels go up, then our hemoglobin saturation, is it going to go up or is it going to go down? So an increase in partial pressure of carbon dioxide means that that tissue is really active. So that means our hemoglobin needs to give up oxygen to it, resupply oxygen to it. So our hemoglobin saturation, the amount that it has, is going to go down because it's giving it off to that tissue. Okay? For pH is that weird scale, right? The small numbers are acidic, the big numbers are basic. So for pH, as pH goes down, little p, big H, when pH goes down, then our hemoglobin saturation is actually going to go down as well. Right? Because it's becoming more acidic, meaning that that tissue is really active. So let's give off some of that oxygen. So the saturation, the amount that it's holding, is going to go down. Okay? That's what you need to be able to describe those relationships that as those tissue levels change, this is how hemoglobin responds. Okay? So there's a question in our exam objectives where it lists some of those different factors and says, okay, what happens with hemoglobin? When this thing goes up or when it goes down, what happens to hemoglobin saturation? Now, I've described one side of that coin. I said when temperature goes up, hemoglobin saturation goes down, but the opposite is also true. If it's cruising through a tissue that is not very active and it's cold, well, you're not doing any work. Why am I going to give you oxygen? You're okay. You're not doing anything. So it's just going to zip on through, and the saturation is going to stay high. It's going to hold on to it instead. So make sure that, right, you understand that the opposite is also true besides those examples I just gave. Okay. Gas transport is what we're talking about. We're talking about oxygen being bound to hemoglobin and how the neighborhood that it's passing through, that environment, can affect whether hemoglobin releases it or whether it holds on to it. Okay, the other thing that we need to talk about in terms of gas transport is carbon dioxide transport. How is carbon dioxide transported in the blood? So we have three different options. The biggest one is it gets changed into bicarbonate ions. That's the main way that carbon dioxide is transported in the blood. A little bit of it, 20%, is bound to hemoglobin, because if we give off that oxygen, now we got a free hand, I can grab some of that garbage for you and take it for you, right? And then a little bit, like 10%, is dissolved in the plasma as well. So we don't change it or anything, it's just dissolved as carbon dioxide in the plasma. This leads us to that equation. You need to know this equation because this is the main chemical way that we change carbon dioxide into bicarbonate ion. Okay? So CO2 plus water, we have water inside of the plasma. CO2 plus water gives us carbonic acid. Carbonic acid very quickly falls apart into bicarbonate ion. So there it is. Now we see the CO2 being transported like that. And at the same time, it releases the hydrogen ion. So that's what makes it a little bit more acidic every time we do that. Okay? The enzyme that does that is called carbonic anhydrase. So you need to know that enzyme, right? Because it's right in the name in terms of carbonic, right? So we can see it has to do with carbon dioxide there. And that is going to do that conversion for us. And the other thing that you should know is that carbonic anhydrase is found inside of the red blood cells. Because it doesn't seem intuitive, because we're talking about carbon dioxide floating around in the plasma as a bicarbonate ion. So why is all this happening, right, in the red blood cell? That's just where that enzyme is going to be located. So that conversion happens back and forth in order to give up or convert carbon dioxide one way into bicarbonate, or the other way from bicarbonate back into carbon dioxide so that we can exhale it into the alveoli and into the external environment, right? So it's a reversible reaction. It depends on whether we are loading carbon dioxide into the blood, or now this blood has gotten up to the lungs, we need to take it out of the blood so that we can exhale it, right? Because we can't exhale bicarbonate ions. We need to exhale carbon dioxide, so we got to convert it back, okay? So carbonic anhydrase is a key aspect to carbon dioxide transport as well. So you need to know where it's transported, mainly as bicarbonate ion. That carbonic anhydrase in the red blood cells is what does that conversion back and forth. Okay? Okay, so let's take a look, go back to our exam objectives and see how we're doing in terms of covering things. So, ventilation, we talked about that, okay? So we talked about the muscles involved, we talked about what they do, we talked about the pressures, the three pressures involved, and how the gradient is generated, right? Because a gradient is the key thing. It has to be high one place, low the other place, that's where you're going to get your movement, okay? 14 was our tracing that we had done on the board before, where we listed out all the anatomical structures that an oxygen molecule would pass as we're inhaling it to get it down to those alveoli in the respiratory membrane, right? So that we did before. We were talking about the vestibule, the nasal cavity, and the pharynx, and the larynx, and the trachea, the bronchi, and the bronchioles, alveolar ducts down to the alveoli, and then across those three layers of the respiratory membrane: alveolar epithelium, basement membrane, capillary epithelium, and then you're in the blood. Okay, so that's just the tracing, doing those things in order. And then external and internal respiration, we talked about that, how that gas exchange occurs. And then, let's see, there's a few other things in there. For this one, this is talking about ventilation. So it goes through, so there's our muscles listed, like we were talking about. Internal intercostals that we see listed there. If we're doing, bless you, if we're doing some sort of forced breathing, right? If we really want to forcibly exhale, we use internal intercostals to really drive the walls of the cavity down. So rather than external intercostals relaxing and they just kind of drift down, we use the internal intercostals and it kind of slams them down instead. As well, you use your abdominal muscles like rectus abdominis, and you really contract those, and that forces the diaphragm up rather than just relaxing and kind of coming up naturally. It gets forced up, it gets pushed up by the organs, the abdominal organs going up against it. So we see that in there. Those different centers that we see there, apneustic center, dorsal respiratory group, and stuff like that, we simplified those into knowing that it is the pons and the medulla, those are the areas of the brain that are in charge of ventilation, okay? So the medulla oblongata has a more direct response in terms of control of breathing, inhale, now exhale, stuff like that. The pons, which is up above it, if you remember, kind of has that supervisory role as well. So it kind of supervises, okay, so when do we change over from inhale to exhale? What if we have to do a really big breathing rate or a big breath in or something like that, a forced expiration? So it kind of handles kind of upper level, if you want to consider them respiratory functions. But you just need to know those. You don't need to know those specific areas that we see listed there. What else do we have there? Oh, so we did have this. So remember we went through spirometry, the different lung volumes and different lung capacities. So make sure that you review that as well. So that's going to look like this, right? So these slides that we see here talk about the different lung volumes and different lung capacities that we have. And you need to be able to name them, be able to define what it is, just a one-sentence description, and then give an average value for it. Likewise, if you were given that graph and it didn't have any labels to it, you'd be able to recognize, okay, which one of those is tidal volume? If it just said A, B, C, D, E, and I said which one's tidal volume, you'd be able to point to that one way over on the left there, that small little hill, and say that's the one that's tidal volume, okay? So you need to be able to recognize it as well as be able to define it and give an average value for it, okay? So those are for our lung capacities. When we get down here, this one is really just talking about, again, what we talked about for gas exchange, PO2 and PCO2, talking about how oxygen and carbon dioxide is transported in the blood. Don't worry too much about the chloride shift. And then this one is what we were talking about already, right? How each of those factors, when they change, how it may affect whether hemoglobin releases or picks up oxygen, right? Whether it becomes less saturated or it becomes more saturated again. And I just noticed when we were going through it early in the week, a few of us were going through this. I apologize, this is like a formatting thing. So 23 and 24, if you read through this is actually just one sentence. It's like I hit the return and automatically added those numbers. But it's to explain how the following affect the affinity of hemoglobin for oxygen, oxygen saturation, and the ability of the blood to load up and unload oxygen. That's like one sentence. It's not like three different objectives. Okay? It's basically looking at those four things and how they affect hemoglobin holding on to or releasing oxygen. Okay? Okay, so three different chapters that are part of this exam. Lymphatic, where we're mainly talking about fluid and this whole other series of vessels, and we need to be able to talk about, okay, what are these new vessels? How are they similar or different to cardiovascular vessels that we saw before? What are they doing? What are they picking up? What's their role? And then a little bit about lymphatic tissues and organs that make up that system. What's a lymph node? What's the spleen do? What does tonsils do? What does the thymus do? Stuff like that. How they contribute to that system. And then we moved into immune system. And immune system, we spent a lot of time with that, right? First line, second line, third line defense, understanding all the different players involved. And then we wrapped up with respiratory system. Again, what are the different organs? What are their role? And then how do these four processes work? Because that explains how this system actually does its job in terms of getting oxygen to the cells, getting carbon dioxide from the cells, etc. Okay? All right. Well, I hope that that review helps to clarify some of those exam objectives as well as some of our major topics. So now, in the remaining days, right, we have our lecture quiz due tomorrow. This is your time to be really going through that. You have clear answers. Now make up flashcards. Make up step-by-step. How do I explain this? How do I explain that? And get studying it. Because now you're clear of what the expectation is. Now you want to focus on that and study it. Get your repetition in so that you can, if you get that question, oh, I know this answer, blah, 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 blah. It comes really easy. Okay? All right, so in between time, got office hours later today. I'll be available by email prior to our exam Saturday night as well. So make sure to reach out if you run into anything along the way and you need help.